welcome to the 2020 Brain and Transcranial Photobiomodulation Virtual Summit. I'm your host, Dr. Joe Doduro. My guest is Dr. Professor Michael Hamblin, who's the principal investigator at the Wellman Center for Photomedicine and at Massachusetts General Hospital and associate professor of dermatology at Harvard Medical School before his retirement. And he's affiliated faculty at the University of Johannesburg, South Africa. He gained his PhD in organic chemistry from Trent University in England. His research includes photodynamic therapy, photobiomodulation, drug therapy, drug delivery, tissue engineering. He has published over 500 peer reviews, articles, 150 conference proceedings, book chapters. He wrote the book on it, Brain and Photobiomodulation. He's editor-in-chief of Photobiomodulation, Photomedicine, and Laser Surgery, associate editor for 10 journals, an editorial board of 20 journals. He has an H factor of 106. He's the #1 researcher cited in photobiomodulation. He's won this award, that award, and the other award. You can go look at it. But he is I want to call him the Godfather. Everybody looks at him that way. And he comes to us, let me show you, from Melrose on the Scottish border there in northern UK. Did I say that right, Doc?
Yeah, well, yeah. Southern Scotland, I guess, yeah.
So we're coming from all over the world here. Thank you so much for your participation. One thing I want to know is, how does a PhD scientist get leaned into photobiomodulation? How did they start? How did you start into photobiomodulation? And then how did you get into the brain photobiomodulation, if you would?
Okay, so I got my PhD in synthetic organic chemistry quite a long time ago, and started doing research. And one of the things I was researching into was porphyrins, highly pigmented molecules that are deeply colored, absorb lots of light. And these are used for photodynamic therapy, which is you excite the porphyrins with light, they produce reactive oxygen species, and they generally kill things. They kill cancer cells, they kill bacteria, sort of viruses even, kill all the nasty things you don't want. But when you do these experiments with PDT, photodynamic therapy, you have to have a control. And the control is light alone. And most people in PDT think the light alone doesn't do anything. They're actually mistaken. It does exactly the opposite of PDT. So instead of killing things, killing all the nasty things, it actually stimulates healing and regenerates tissue. So that's caused quite a bit of consternation in PDT folks, because they say, my light alone controls are doing all this stuff. And you have to bear in mind that quite often it's red light that PDT people use. So obviously you can't ignore this. You have to sort of investigate it a bit. And that led me into photobiomodulation.
I see that. And then how did you go from, how did you get to the brain, if I can ask? How did they, you've been doing it on all the tissues since when? When did you start doing that? Right from the beginning you were studying light.
Yeah. Well, once you start studying photobiomodulation, a lot of the things people study are like healing injuries, healing wounds, reducing pain, reducing inflammation. But obviously, for a long time, people have been studying the effect on the brain. And when you look at the mechanisms of the effect, you quickly figure out it's to do with the number of mitochondria in the tissue. And tissues like skin for wound healing actually don't have many mitochondria. And the organ in the body which has the most mitochondria per unit weight is, of course, the brain. There are other organs like the liver, the heart, and the kidneys that have a lot of mitochondria, but it's not so easy to get light there. So the effects of photobiomodulation on the brain are probably the most pronounced of any part of the body. not only that, of course, but things going wrong with your brain is of huge interest to society and medicine. Everybody's worried about, their brain function as they get older, their memory, their mood, neurodegenerative diseases, strokes, traumatic brain injuries. There's a bucket load of disorders and diseases that affect the brain, and people are really worried about it.
Amen. And so you're going to shine the light, as you like to say. You're going to walk us through your stuff. Would you like to share your screen, show your slides?
Sure. Do I?
Go ahead.
So this is my contribution to the photobiomodulation of the brain symposium. The title is Transcranial Photobiomodulation. Can be used for traumatic, degenerative, and psychiatric disorders of the brain. So it's pretty broad spectrum treatment, if you will. If we can move. I think go one by one, hopefully. So here's an outline of the talk. My lab has worked a lot in mechanisms over the years, so I think I'm considered one of the world authorities in the mechanisms of photobiomodulation, so we'll talk a bit about that. Photobiomodulation for traumatic brain injury in mouse models. This is how we got started doing laboratory research in the brain, was in TBI, and then I'll talk about a lot of clinical collaborations that I've had for TBI in humans and patients, Alzheimer's, Parkinson's, Gulf War illness, psychiatric disorders, and also ask whether you can use photobiomodulation for cognitive enhancement. So getting started with the mechanisms, as I said, the main chromophore in tissue for red or near infrared light is the mitochondria. And the principal molecule within the mitochondria that absorbs the light is this guy called cytochrome C oxidase, which is a complex protein, has 13 separate subunits. It has 2 metal centers, 2 coppers and 2 hemes, each of which can have a different oxidation state, oxidized or reduced. So if you do the math, you'll see there's 8 different redox states of cytochrome C oxidase, and every one has a different absorption spectrum. Not very different, but, measurably different. And when you shine the light, and the wavelength you choose, is to some degree governed by the redox state of cytochrome C oxidase. But basically, it increases the activity of the enzyme. So one hypothesis is that you photodissociate nitric oxide, which is the little NO you can see there, this little orange guy that's binding to the copper and inhibiting oxygen binding, inhibiting respiration. It's not actually proved that you dissociate this, but it's a nice theory. But it is known that oxygen consumption goes up, mitochondrial membrane potential goes up, ATP goes up, because that's the purpose of this whole electron transport, is to make ATP. You can measure NO released, which led to this hypothesis that you're dissociating it. There's a brief burst of reactive oxygen species, and there's significant changes in calcium. One important change in the mitochondrial metabolism is that glycolysis is switched to oxidative phosphorylation. And this turns out to have quite an important consequence, which I'll probably get onto later, I think. So you've got these changes going on in the mitochondria, the nitric oxide, the ATP, and the ROS, and there are several important transcription factors. AP1 is a June-Phos heterodimer, which is activated by cyclic AMP. Protein kinase D is activated by the brief burst of ROS that will cleave I kappa B, thus allowing NF kappa B to go to the nucleus. And these are highly pleiotropic transcription factors. They will activate maybe 100 or more genes that have AP1 and NF kappa B response elements. Not only that, but nitric oxide can also trigger a lot of signaling pathways. So all these signaling pathways in gene transcription lead to production of growth factors, extracellular matrix, cell proliferation, anti-apoptosis, and pro-survival. So a relatively brief exposure to light can affect cells and tissues for a long time. And even as long as a month. We have data in mice that a single exposure to light can last for a month. As I said, the two major effects of this switch from glycolysis to oxidative phosphorylation. The first one is activation of stem cells. Because stem cells live in a hypoxic niche, they carry out glycolysis because there's no oxygen. But when their mitochondria are activated, they leave their niche in search of oxygen, and that activates the proliferation and differentiation programs in the stem cells. So it really mobilizes the stem cells from their niche, and quite often that's in the bone marrow. The other consequence of this switch from glycolysis to oxidative phosphorylation is that macrophages have an M1 phenotype of pro-inflammatory and carry out glycolysis. However, when they activate oxidative phosphorylation, they switch to the M2 phenotype, which is the anti-inflammatory phenotype. So this M1, M2 macrophage polarization phenotype is a really hot area at the moment, and people are studying it with many, many applications. It's important to realize that in the brain, the M2 macrophages can actually phagocytose stuff. And as you know, many problems in the brain arise because you get aggregates of proteins, and amyloid plaque is a famous one, but alpha-synuclein, Huntington, there's many, many protein aggregates you can get in your brain. And if you want to get rid of them, you want to switch your microglia to the M2 phenotype, and photobiomodulation just happens to be able to do that. There are other mechanistic pathways of photobiomodulation. One interesting one is this idea of heat and light gated ion channels. So these are TRP ion channels, transient receptor potential. They're activated by two kinds of light, blue and green light. They have an absorption band in the blue and green due to their chromophore, which is a 13 cis retinol. This is bound to proteins called opsins, and when this switches from cis to trans, because it absorbs the light, it opens the heat and light gated ion channels and calcium gets in. There's also heat gated ion channels, and the hypothesis is that some longer wavelengths in the near infrared, such as 980 nanometers, for instance, can heat up nanostructured water, which is not bulk heating. It's a very nano-scale heating, just enough to make a conformational change in the ion channels. Again, calcium is the main mediator. Now, the last hypothesis relies on, again, on this water being a chromophore. The idea is that interfacial water in the mitochondria, its viscosity is lowered when it absorbs red near-infrared light. And because ATP synthase, unit 5, has a little molecular rotor, if you reduce the viscosity of the water, it'll turn faster. So folks like Andrew Sommer in Germany believes that's one reason why you get extra ATP with photobiomodulation. So recently it became clear that normal blood contains circulating cell-free respiratory competent mitochondria. This was literally towards the end of last year, right? Now you might well ask yourself why, when people have been studying blood for a century or more, nobody has ever noticed that there's a large amount of circulating free mitochondria. Does the blood we know so well contain elements that had been undetectable until now? The answer is yes, according to these folks. So here is the idea. You've got all these free mitochondria in the blood, and they can be activated by light. So this is one reason why when you put light onto the body, regardless of necessarily which part of the body, there's always a lot of blood flow in the skin. And this has these cell-free mitochondria that get activated in, in the same way as the mitochondria in the tissue, such as the brain, for instance. Certainly, as time goes on, the systemic effects of photobiomodulation are becoming more well accepted. One of these reasons for this is that the whole field is moving away from what used to be laser therapy, when people had a focused laser beam and they shone it as a point on a joint or a wound or something, into large area LED arrays. And large area LED arrays certainly deliver a lot more systemic photobiomodulation than a focused laser beam. And so that's one.
Thank you for sharing that reference, because they've been talking about it in the program, but no one has ever did not throw that reference up. There's a lot of stuff in the blood that they haven't been able to classify. It's got to be literally, pretty obvious, but we can't see it. It's the same
right.
thing.
I think this idea that we're cell-free mitochondria is important, because previous to that, we'd sort of assumed it was platelets, because platelets have a lot of mitochondria. But it wasn't exactly clear why activating platelets would produce all these effects.
Correct.
So I think the cell-free mitochondria is a much better explanation than platelets. So this is just a schematic showing all the different parts of the body, diseases, disorders you can use photobiomodulation for. As you see, it ranges from the tip of your head to the tip of your toes, and includes a lot of serious things. We're talking about the brain today, but heart attack, lung inflammation, very relevant for this whole COVID crisis. Kidney failure, diabetes, obviously arthritis.
What's the difference between the blue arrow and the red arrow? Do you recall?
No, I don't. Not really.
No.
It must have been originally a difference, but
yeah, got you. It's okay.
As I say, the stem cells, people like Uri Oron in Israel will shine light on the bones to mobilize the stem cells. And he'll do that to treat the heart and the brain and the kidney by shining light on the legs, which is sort of counterintuitive. But once you realize you can mobilize the stem cells from the bone marrow, and also wherever you shine the light in the body, you will be irradiating the blood by definition. So I think this is one of the key issues in photobiomodulation. To what extent is the organ in the tissue you are shining the light the important thing, and to what extent is some systemic effects involving stem cells and blood irradiation important.
So one of the things that we brought up is a direct effect, which is a lot of times what people are studying in vivo. I'm going to see if I put the laser on the kidney, and then I want to study what the kidney is doing direct. But the indirect is everything else that's getting hit by the light, and that is difficult to kind of study. It's an after effect. So direct means you're shining it right there to study that effect, to have that effect, but the indirect effect is a confounding factor.
And remember, when you put the light on the head, first of all, it has to go through the scalp, and there's a lot of blood flowing in the scalp. And then it has to go through the skull, and there's a lot of bone marrow in the skull. So by definition, before the photons get into the brain, they have to go through the blood and the bone marrow. So the stem cells are found most proliferatively in thin, flat bones, like the hand, the tibia, the sternum, ribs, and you're saying it's even in the skull. Then I know it's in the the small cribriform plate of the nose, but I didn't realize that it was in the thin bones of the skull. Oh yeah, I believe there's quite a lot of bone marrow in the skull. You know how you measure it comparatively, but certainly there, yes. Okay, so moving on. Strokes and TBIs, localized ischemic brain damage. There's been a lot of studies of drugs for stroke and TBI, and by and large, they've virtually all failed. Okay, it's embarrassing that billions of dollars that have been wasted in failed trials for stroke and TBI drugs. Some physical therapies such as hyperbaric oxygen and hypothermia, brain cooling, some of these have an effect, but by and large, the therapies are really not very good. So we originally hypothesized that photobiomodulation could improve TBI by a neuroprotective mechanism. So the idea was that you would inhibit apoptosis, you would inhibit excitotoxicity, and you would be pro-survival. So when you have a head injury or a stroke, as you know, there's an initial damage to the brain, and then it spreads and expands to affect more of the brain. So doing this neuroprotection would be a good way of preserving brain function. And we did some studies originally with different wavelength lasers in mice, and this was a closed head TBI model where basically you do a weight drop onto the head of the mice to produce a traumatic brain injury, and we put the same power density and the same fluence of these 4 different wavelengths, and we found something quite interesting. The first one we tried was this 660, and it worked like a dream. A single exposure, 4 hours after the head injury, and then you follow these mice for a month, right? A single exposure, 4 hours after the impact. Then we tried 810 nanometers, and that also worked like a dream. Single exposure, 4 hours, and you follow the mice for a month. So we thought, both these wavelengths seem to be working fine. So we managed to get a 730 nanometer laser, and we were relatively pleased to find it had no effect at all. Because, being proper photobiologists, you want to see an action spectrum. So you don't want to see every single wavelength of light you use have the same effect. And then we had a 980 nanometer laser, and that also had no effect. But it's interesting that these error bars are actually quite big. And I believe you could get 980 nanometers to work, but the dosimetry is different. remember to compare these four different wavelengths, we use the same dosimetry. So then we were quite excited by this. We moved on with a different model of TBI, which is controlled cortical impact. So you actually have a special computer-controlled machine to make the impact in the mouse brain, because doing a weight drop is okay and it's clinically relevant, I mean, but it's not the most reproducible way of creating a brain injury. So there's actually a company that makes a machine to do traumatic brain injury in mice and rats. And we got this machine, and we chose to use 810 nanometer laser, and we wanted to compare pulsed wave with continuous wave. So again, a single treatment, 36 joules per square centimeter, 50 milliwatts per square centimeter, and it's either pulsed at 10 hertz, 100 hertz, or CW. One treatment, 4 hours after the TBI, follow the mice for a month. All of these laser treatments worked fine, as we'd shown before. It'd be surprising if they didn't work fine. But what was interesting is that the 10 hertz was significantly better than the CW or the 100 hertz, which are here. So this 10 hertz one was doing something magic. Quite what is doing this magic, we're not exactly sure, but you know, the prevailing hypothesis at the moment is it synchronizes with the alpha rhythm in the brain, which is about 10 hertz. So Lou Lim has done a lot of work with different pulse structures synchronizing with different brain rhythms. So he has alpha, which is 10 hertz, he has gamma, which is 40 hertz, and he even has other pulse frequencies now. The idea is if your pulse synchronizes with an intrinsic rhythm in the brain, it'll work better.
Matt, can I ask one little question about that? Can you go back one slide?
Yep.
Now, if you have a damaged brain and it's got that neuroinflammation cascade that you talked about, what would be the difference between into the brain between a constant wave and a 10 hertz? How does that change that inflammatory factor? How does that change the mechanisms that you talked about? Do you have a clue on that? Because I don't.
Well, you know, if you believe the work from MIT where they showed, not with photobiomodulation, was pulsed blue light, but pulsing it at 40 hertz does this microglial phenotypic switch. So it switches the microglia from M1 to M2, which is clearly important, and that is anti-inflammatory. As I say, they did 40 hertz, and they showed 40 hertz was better than 80 hertz, or I think 20 hertz. So that is one idea. Somehow this frequency in the brain does appear to affect the microglial polarization phenotype.
Excellent.
Okay, so what we'd shown there was a simple neurofunctional testing. Could the mice walk in a straight line, balance on a beam? It didn't really test cognitive function. So then we thought, well, can we affect learning, memory, and also these neuroprogenitor cells, which some people call neural stem cells. But anyway, they are the cells that will help the brain to repair itself. So this was the Morris-Water maze, which is the test for memory and learning in mice. You know, they have to memorize from the visual cues where the hidden platform is, and they have to see if they can swim to where it used to be.
Mm-hmm.
So here we have normal mice, sham injured mice, TBI mice takes them longer to remember where the hidden platform used to be. And with 1 laser treatment or 3 laser treatments, they have significantly faster swimming to where the platform was. So first of all, they have to remember where it was, and then they have to navigate with the clues. So one way of looking for these neuroprogenitor cells is to inject in the mice BRDU, which is a nucleotide precursor that gets incorporated into DNA. And you can then stain with an antibody that recognizes this BRDU. And what this shows you if cells are dividing in the brain, okay? And mostly cells do not divide in the brain. There's only a very few cells that are capable of dividing, which are these neuroprogenitor cells. And you can also double stain them with N to show that they're neurons. And these are these green cells here, and with the 3 photobiomodulations at 7 days was quite a good increase. Not so much at 28 days, but definitely at 7 days. In addition to these neuroprogenitor cells, another very important thing you can have in your brain is BDNF, brain-derived neurotrophic factor. And that is related, A, with this neurogenesis, the neuroprogenitor cells, and B, with this synaptogenesis, which is the formation of newly formed connections between existing brain cells. So for your brain to repair itself, A, it's a good idea to have some newly formed brain cells, but B, it's also a good idea to have newly formed connections between existing brain cells. So this is staining for BDNF. Again, there's not a huge amount of it. It's this red color here, and you see a nice increase with the 3 lasers at 7 days in both the subventricular zone and the dentate gyrus, but not again at 28 days. And this is staining for synapsin 1. So synapsin 1 is a protein which is formed in these new connections. And interestingly, you see it's upregulated at 28 days, not 7 days, 28 days, and in different parts of the brain. So it's much more in the cortex, the new connections, whereas the BDNF and the progenitor cells were in the subventricular zone and the dentate gyrus. I mean, there's a little bit here in the subventricular zone.
Okay.
So that's where you want the new connections in the cortex, because that's where you do your thinking. So we changed our overall hypothesis a little bit. Now, I'm not saying we don't do neuroprotection. I'm sure we do, and other people have shown it. But we do think that we're helping the brain to repair itself by stimulating neurotrophins, synaptogenesis, and neurogenesis. Again, there's all these other good things, the anti-inflammatory, the antioxidants, pro-survival, angiogenesis, lymphatic drainage, increased blood flow. So the beneficial effect in the brain, multifactorial, and for any given brain condition, it's not immediately obvious which of these is predominant. I mean, they're all good, but you could say, okay, for brain injuries, clearly repair is important. But, for neurodegeneration, for Alzheimer's and Parkinson's, helping the brain to repair itself could be the critical thing. Because you're not really going to stop the overall degenerative process, right? By and large. So that's all the laboratory studies. The next things I'll be talking about are clinical studies, which are mainly done with collaborators. So this was an initial collaboration with Marnie Naser, again from Boston, who treated 2 case reports, patients with who'd had head injuries. One lady, 7 years after a traffic accident, she had real difficulties for 7 years after a crash, but Marnie treated her initially, 8 weekly treatments, and then she got a home unit and she's used it daily for 5 years. And it's this MEDX cluster, LEDs, 870 nanometers, 633 nanometer LEDs. Another lady had multiple concussions, PTSD, treated with a home unit, memory and executive function tests improved by more than 2 standard deviations. And she could go back to work, which, because having a chronic TBI can destroy your life. There are people who've had virtually their entire lives destroyed because they were unlucky to have a serious head injury. This is some of the psychological tests that Marnie did. The Stroop test, this is where you have the name of the color and the color of the font differently. So you'll have the word red and blue font and green and red font, and it's really quite confusing when you have to do it rapidly. This is stories, immediate recall, and delayed recall, 30 minutes. And again, significant improvements over what they were before, pre and post LED. So Marnie was very excited by this data. Okay, only 2 single patients. So what she did then, she gathered together a case series of 11 patients. And out of these 11 patients, you will see that 1, 2, 3 didn't really have any benefit, but the other 8 out of 11, had significant benefits. Someone could go back to work. One couldn't talk, but became quite verbal. They could do all their business. So it made quite a big difference to these folks' quality of life. So Lulim in Toronto with Firelight has a small clinical trial for Alzheimer's. It started off, funnily enough, as a randomized controlled trial, but turned out that, these are old folks that were brought in by their carer, and the ones that were getting the sham placebo treatment didn't get any better. Okay, but it ran for 12 weeks, and they all dropped out. The ones that were in the sham group all dropped out. So the only ones that finished the trial were getting the real PBM. And this is the MMSE, Mini Mental State Exam, and this is the ADS COG, the Alzheimer's Disease of America Cognition Score. And as I say, they were treated twice a week for 12 weeks. And this is a remarkable improvement. So just to give you a feeling of the size of this improvement, it was 7 times bigger than the improvement in the large trial for Aricept. So Aricept is an acetylcholinesterase inhibitor. It's one of the few drugs approved for Alzheimer's. And, they did a trial with hundreds or thousands of people, and the mean improvement was only a tiny fraction of this. Okay. So when the treatment was stopped after 12 weeks, they deteriorated. As I say, Alzheimer's is a progressive degenerative disorder, so you're probably not going to cure it forever with photobiomodulation. But it's not on this graph, but at the end of this part of the study, so this was when they were treated, this was the follow-up, but again, they were given a home use device, and they all improved for a second time. Because you have to sort of give these, but they did so well, you have to give them one, basically. Other folks have repeated this type of result with dementia. I mean, it's people are loath to say they have Alzheimer's because you really need sort of pathological proof we've got Alzheimer's. Everybody knows that old folks quite often get this dementia, right? So again, ADS-COG, another performance score, and the ones that are getting the photobiomodulation improve and the controls deteriorate. So that's similar to what Marnie find. If you don't do anything, they get worse. If you do something, they get better. But Linda had some neuroimaging studies, arterial spin-labeled perfusion imaging, and can show that you get improvements in the neuroimaging in these folks, which is important because people like to see something that's not just, a psychological scale, basically. This was a single case study from, we are on this, Joe.
Both of us.
From Farzad. And you made these devices, I believe.
Correct.
So here they are. Interesting set of 3 devices, one for the head, one for the body, and one for the nose. And I think that's a reasonable thing. Get as much light into the body as you can. This person, only a single case, but he improved a lot. Quite a big, very big differences here.
I can speak to this. Since I did the calculations. So the point is that the changes were noticed almost immediately, and that it went from an 18, which was a mild dementia, to 24, which is practically near normal. Memory improved. The first time it was rated that the smell or olfaction returned to a baseline, saying that we did get something in there. But the other thing is we looked at the caregiver scores. Not so much as what does the caregiver think about the patient's change, but what was the self-care, and then how much burden was there. So these were significantly improved. So it's a functional. We had a memory, we had some neurophysiologic, and we had the caregiver stress went down. The point is, it was home care, totally home care.
That's what Lou Lim said, that when these folks were given their own home use device, their caregiver used to use it as well, because they could see what a big effect it had.
Of course, we needed caregiver stress is real. That the caregiver is the diagnosis with no treatment. So that's why moving them around was difficult. Thank you for that.
So this is Martin Berman's thing with the Cognito Light helmet, which is not the most artistically appealing helmet, but it does the job. It's got 1,070 nanometer LEDs in it. And again, folks with dementia patients testing memory and cognition. A lot of these things have significant p-values. Not all of them, but some of them. Various cognitive tests, basically, but also cortical perfusion measurements to back it up. In Australia, they've been doing Parkinson's disease. And Parkinson's is, you can get dopamine replacement therapy, hydroxydopa, but they say it just treats the symptoms, and it's a progressive neurodegenerative disease with a lot of mitochondrial effects. So not well treated at all. So Catherine Hamilton in Tasmania came up with these light buckets. So the idea was you could buy at the local hardware store, you could get these LED strips, you could wind them round inside a light bucket, 4 wavelengths, 6, 78, 58, 10, 9, 40, 20 to 30 minutes twice daily. Again, you know, this is not a controlled trial. It's just 4 patients, basically. But these are all the symptoms of Parkinson's. And the way this is set out is if something is red, it got worse. If it's yellow, there was no change. If it's green, it got better. So you see a lot of green, a few yellows, and no red at all. And, you know, Parkinson's, it's a degenerative disorder. You expect things to get worse. So the fact that virtually everything got better or stayed the same is amazing. You know, handwriting is one of the big things in Parkinson's, is so-called micrographia. So your handwriting gets really tiny and nobody can read it. So that improved. So these light buckets, they're quite keen in Australia. They're doing bigger trials now. As is Lou Lim for Alzheimer's, he's doing bigger trials. Everybody who gets good results starts to get funding to do trials, but it's taken them a few years. You know, it's not like it happens overnight. Marnie's also done Gulf War illness. So as you know, Gulf War illness is a big deal in the US, and the rest of the world doesn't really think it's anything, but in the US it's a big deal. And they use the Violite Neuro Alpha 3 times a week for 12 weeks. Again, Gulf War illness is not just a cognitive disorder, it's a systemic mitochondrial disorder. So you will have fatigue, muscle pain, digestive symptoms, all sorts of things. But you know, if you look at this, a lot of them improved. And it's pain. And Gulf War illness is a lot of chronic pain. But so the sleep gets better. And this is one of the big things that you notice with brain photobiomodulation, is everybody sleeps better, basically. So this was Fred Schiffer, who some time ago we did this study for depression and anxiety. It was a single exposure to the head with Hamilton Depression and Anxiety Scale 2 and 4 weeks after a single treatment with this little near-infrared LED device on the head. And what you find is that 2 weeks after the single treatment, virtually everybody improved with both the depression and the anxiety scale. What you see here is objective measurements of cerebral oxygenation and blood flow in the brain. So this is actually objective measurements, but this, of course, is scales. Paolo Casano, obviously, being a psychiatrist, was very interested in this, and he started, this was the Photothera Laser, the company that did the stroke study, who actually unfortunately went bust, but never mind, it doesn't mean photobiomodulation doesn't work just because Photothera went bust. And he treated 6 times, not just once, 6 times, a major depression, and these folks' mean depression scores steadily improved and carried on improving after the end of the 6 treatments. So there's quite a lot of devices people are coming up with for this, putting light on the brain. These are just published ones, but there's several other light-emitting helmets that people have got now. And this is one from China. You can order it. Could order 500 pieces a month of this near-infrared helmet. It's, I think it's about $1,000, something like that. That's sort of equivalent. But, because it's such an attractive thing for LED companies to make, I think quite a few are making these helmets now. This was autism spectrum disorder. It's a poster, so I'm not going to go into it in any detail. It's a bit difficult to see, but basically this is young kiddies with autism and putting near-infrared light on their head, and it has a big effect. I mean, it's quite dramatic, actually.
Paolo Cassano is the first one to even kind of observe this enough to gather 6 people and actually do it.
Yeah. I think because autism is, the sort of thing parents really worry about, anything you can give these kiddies to help them. Historically, people have been a little worried about treating kiddies in case you do some permanent damage, but I think now it's realized that near-infrared light is not going to do any permanent damage, and you're probably going to help them quite a lot. So Agnes Chan is in Hong Kong, and she's done sort of cognitive enhancement type stuff. So this was the first one, was cognitive function in older adults. So these are people who have not been diagnosed with anything, right? They're just getting old, and there was 2 tests here. One was the category fluency test, the Erickson flanker. This, whereas you just have to say that this one doesn't match and this one does. So again, it's a computer thing. You have to press the keys on the keyboard quickly. And there was a significant effect there, but the really interesting thing was this category fluence test. So this is like, how many animals can you name beginning with B within a certain time, right? You say buffalo, bison, whatever. The ones that got the near-infrared light on their head could name like 3 times as many in the category in the time, which is astonishing.
That's memory and recall and all that good stuff that they grabbed.
Yeah. So this is another one from Agnes. Neuromodulation makes a difficult task less arduous. So real or sham, single photobiomodulation, 800 nanometers, 8 minutes, 28 joules. A sort of standard sort of dose to the head and the forehead. This is another cognitive task where you have to think back 3 goes ago. So a 0 back is easy, you just have to think for the previous thing, but a 3 back you have to think for the one that you heard 3 goes ago, which is actually not easy.
Not easy. And
the hemodynamic responses were measured. So this had both the LEDs and the near-infrared diodes to measure the hemodynamics in a same sort of headset. So here's the LEDs here that are doing the therapy, and here's the sources and detectors for the near-infrared spectroscopy. There are only minor changes in memory and reaction time, nothing to speak of, not really significant. But the interesting thing was that there was significantly lower blood flow. Okay, so the people performed the same on the cognitive test after the photobiomodulation, but they needed a lot less cerebral blood flow to do it.
All right, Mike, I want to talk about this for a second, if you don't mind. What is your take on this? Because, we've seen everybody say that we had increase in blood flow when we do photobiomodulation. We see increase in, these type of activities, but these showed, what we're looking at is oxygenated hemoglobin. So this is saying that less oxygen was delivered or less oxygen was used, or what is it saying? Do you know?
Well, I believe that the amount of oxygenated blood delivered to parts of your brain is very closely regulated by the amount of neural activity. So if you think of something in your brain, immediately it'll light up on a fMRI. And, this is the whole rationale of doing fMRI, and they look at all the different parts of your brain that light up when you're doing different cognitive tasks. So it's remarkably rapid, and to get the same amount of cognitive performance with less oxygen consumption is showing that something's happening more efficiently. At least that was Agnes's interpretation. It's interesting anyway.
Yes, definitely. It had an effect. It had an effect.
So this was in folks that were diagnosed with mild cognitive impairment, 3 case reports. Again, the same sort of photobiomodulation device on the forehead. Post-stimulation, some of them were, the visual was a big improvement. The verbal maybe not so much, but certainly the visual memory was a big improvement. You know, a lot of people say, well, is this photobiomodulation? How does it compare to other non-invasive brain stimulation techniques? And this is just kind of, you know, you've got your deep brain stimulation where you put an electrode in the brain. Some people want to put an optical fiber into the brain. Clearly repetitive transcranial magnetics, that's a big thing these days. Transcranial direct current where you put the electrodes on the head. And low intensity focused ultrasound. So there's all these different ways of non-invasively stimulating the brain, and to some degree they're used to treat similar sorts of disorders: Parkinson's, epilepsy, TBI, depression, addiction, cognitive enhancement. But I don't think they're the same mechanism. I mean, okay, they're similar, right? Non-invasive brain stimulation, but you know, you don't get action potentials with photobiomodulation the same way as you do with TMS. But it's sort of interesting, and it's pointing to the, you know, the poor performance of brain drugs, right? Whether they're for psychiatric disorders, for neurodegenerative disorders, most of the drugs people try and give you to help the brain are disappointing. Side effects are addictive. They really are not a good idea. So this was the paper I mentioned from MIT, where they originally started to do optogenetics in mice. So they put a fiber optic into the brain, pulsed at 40 hertz with blue light, and they found that Alzheimer's transgenic mice had a marked improvement, had less plaque. And then they also flashed the blue light at the mice. So first of all, they put a fiber into the brain for optogenetic mice, and then they took non-optogenetic mice and flashed light from the outside. And in both cases, the 40 hertz was better than random 20 hertz or 80 hertz. So this 40 hertz frequency, so it attenuates amyloid load and modifies microglia. So that's thought to be the mechanism of action. So this is just kind of summarizing the whole thing. There's all these things that go wrong with your brain. A lot of them are traumatic. We talked about TBI. There's also stroke. A lot of people have done stroke, but also global ischemia. If you have a heart attack, you know, if your heart stopped for 8 minutes, you stand a good chance of having brain damage. So the earlier you can get the near-infrared light onto the head of somebody who's had a heart attack, the better. Birth trauma, a lot of babies get brain damage during birth because they're starved of oxygen. You know, a few people have shone the light on the head of people who are in a coma. It's not like they wake up, it's normal, but it definitely has an effect of people who are in a coma. A lot of psychiatric disorders. Fred Schiffer's now studying addiction, how to say sleep, depression, anxiety, PTSD. neurodevelopmental, autism we mentioned, ADHD, and then a whole bunch of neurodegenerative diseases. Obviously Alzheimer's, Parkinson's are the big ones, but ALS, frontotemporal, vascular Lewy body, PPA, CTE from the football players, mad cow disease and Huntington's. All these are progressive neurodegenerative disorders where helping the brain to repair itself could actually make a big deal. So conclusions, I'm going to read through these. Pretty much all the studies have tended to be successful. I mean, that's a generalization, but the number of studies that have been negative is remarkably few, okay?
I'm with you.
That's
a squad.
This was my lab for a few years ago, but as I say, I've sort of retired now. I haven't given up working. I've just moved back to the UK, so my lab has dispersed itself to pastures new, and we have funding again from various folks.
Nice. Look at all that. I see Kleber in there.
There's Kleber, yeah.
We've got a nice squad. We'll have more of those people as we progress and keep going. And thank you, Doc. I have a couple questions, if you don't mind.
Not at all.
Do you want to take a sip of water while I?
Okay.
Is that apple juice or beer?
That's beer.
Right. Well, drinking on the job has never been easier, right?
Right.
So let me ask you about this, the ROS burst that happened. When you read about mitochondrial activity, if you have an increase in ROS, it's usually bad.
Yep.
So how do you kind of say that the theoretical effect is that we're going to have an increase in our ROS as we do this, and that's going to create a good thing, a good outcome? How if you're going to...
So it's kind of well known that small amounts of oxidative stress are good for you, right? I mean, it's one reason why exercise is good for the brain. And there were a lot of studies 20 years ago where people tried to give antioxidant vitamins for all sorts of diseases, cancer, heart disease, brain disorders, and antioxidants were the big thing. Now, every single trial with antioxidants failed, every one. And the reason they failed is that small amounts of oxidative stress are actually good for you. They stimulate a lot of signaling pathways, stem cells, regeneration, pro-survival mechanisms. And the reason a lot of these so-called resilience pathways or hormesis pathways, preconditioning, why a lot of these things work by producing a small amount of stress to activate protective mechanisms.
Thank you for making that clear. Now, you went through a lot of the mechanisms as the nucleus, as the mitochondria talks to the nucleus, and then the down genetic transcription. But, we've had some people talking about the microtubule activity. And, it's how that's sort of the structure between the mitochondria and the nucleus and the whole cell. You want to bang around a little bit of your thoughts on the microtubules and what happens when we put light on the brain or the body or whatever?
Yeah, I'm not personally convinced the microtubules are particularly important. I know there is a theory, but part of this theory is all tied in with biophotons. Okay, so biophotons are naturally occurring photons emitted by cells, but they are ultra weak. And when you say ultra weak, we're talking 1 billion times weaker than visible light.
Billion times?
1,000 million times weaker. So a lot of them are single photon things. You need a single photon counter to measure them. So nobody's saying these aren't produced, and they are related to the mitochondrial activity of cells for sure. But the problem arises when you want a biological detector for them. Because I say the actual detectors in the lab are single photon counters that cost tens of thousands of dollars because they're cooled to absolute zero almost, right?
Uh-huh.
It is difficult to envisage how a biological single photon detector could operate. In fact, personally, I think it's impossible. But so the folks that talk about photons being transported through microtubules in the brain is a sort of interesting idea. And, it's even one basis for the foundation of consciousness, for goodness sake. But I think it's a little bit like science fiction.
I like it. So how about this, though? So by the bioemission or the bioluminescence, what do you say? Because there's a couple things. You did a great explanation, and I don't want to get the words messed up because I don't want to be like the U.S. president who says go out in the sun. But you made a great analogy that standing in the noonday sun for 20 minutes gives you I don't want to mess it up but like 5,000 joules, which is like more than anybody, all the trials to get. Do you remember that?
That's a lot more than that. I mean, if you lie in the sun at noon, near the equator, right, you cover yourself with sunscreen, you lie in the sun for an hour, you will absorb 1 million joules, not 5,000, not 50,000, 1 million. Okay?
Yeah.
Now, it's not going to do you any harm. It may be more than you need. Who knows? One thing to realize is that everybody's different, right? And some people are very sensitive to light. They're not common, but other people are very insensitive. So they're basically like blocks of wood. You can shine light on them all day and nothing much will happen. Most people are in between, so-called bell curve. So everybody wants to have a standard dosage of light, but there's no such thing. I mean, some people, a little bit of light is all they need, and some people need it for half an hour or more.
So let's talk about the two different levels that I want to talk about. So what about these free mitochondria? What about these free-floating exosomes in the blood? And what about the ability of these tissues that are going in the plasma, for lack of a better term, being excited and then downstream releasing that energy? I mean, if you got a lot of that stuff and they're picking up a lot of light, and you know what I mean? If they get excited, you're the one that explained that the valence goes up and the valence goes down. What about that?
Yeah, I'm not sure that exosomes have chromophores in. I mean, exosomes have a lot of different things in them. I'm not aware of them having chromophores, but the free-floating mitochondria certainly do. And we don't know yet what they're carrying around. I mean, obviously mitochondria do signaling. So presumably they are carrying around signaling capacity.
Mm-hmm.
And that's still a hypothesis.
So what we're saying is that there's two different things that we might be looking at. One would be some type of internal the body gets energized and the body lets go of the energy. It's got to go someplace, right? Just doesn't just decided in some sense. It can turn into ATP.
Yeah, a lot of people have this idea. There's no photobiomodulations like photosynthesis. The cells absorb energy. I mean, it's not the same at all. Photosynthesis, the energy of the photon is directly translated into a chemical reaction, right? In photobiomodulation, the energy of the photon alters the activity of an enzyme, which is a different thing.
Very different. Well, it makes it more efficient. So let me ask you this then. What is your thoughts about physical properties of the photon being able to change and bang into the membrane and the receptors and all the stuff that's out there. Any thoughts on that, that there's a physical property?
This is the whole idea of nanostructured water, because water is by far the dominant chromophore. And Jerry Pollack was the one who figured out that nanostructured water was very different to bulk water. It had different physical properties, different absorption spectrum, and it could change its properties when it absorbed light. So this is the key thing. But you only get nanostructured water in hydrophilic, hydrophobic interfaces. So it's very close to cell membranes and specifically in intracellular organelles, especially the mitochondria.
Here you go. So it's a key.
So this is probably what you mean by photons banging into membranes, I think. The idea is that, you know, if this interfacial water can absorb even a small amount of energy, it can change its conformation, or proteins in it can change their conformation.
Thank you. Now, you mentioned one of the things that you saw or that you postulated was an increase or a change in lymphatic drainage, you know, with photobiomodulation. Where's that written up? Who's talking about that?
Well, you know, several people are talking about it. So photobiomodulation is a treatment for lymphedema, which is a common complication of cancer surgery. And in Australia, they're quite keen on this photobiomodulation for lymphedema. And the idea is because it's well known you get increased blood flow in capillaries and venules, that lymphatic vessels have similar physiology. So nitric oxide is a vasodilator and a lymphodilator. So if you're happy with increased blood flow being due to nitric oxide, it makes sense that increased lymphatic flow will also be.
Good call. So while we were sitting there and we've looked back at the 20, 30 years of your research, and we're going to be turning the lens the other way now. We're going to be at the 2030 Brain and Transcranial Photobiomodulation Virtual Summit, and you could still be the keynote speaker. What do you think the future is going to hold? What do you see happening 10 years from now? What's going to be available for the public? What's going to be the research going to be looking at? How do you see it playing out?
Well, yeah, I think folks are going to have an LED helmet at home as they get older to help them sleep at night, to help their memory. You know, folks who are actually suffering from a disease should, whether they're going to be prescribed one or they go to somebody's office and have the helmet put on them. But one of these things is going to happen because there's no side effects and it seems to be highly effective, and it's a big deal. People are worried about brain disorders. Personally, I think it's more likely that, you know, they'll be mass marketed almost. The companies are, a lot of companies selling them now. I think the only reason people hasn't taken off is people haven't heard of it. And if they have heard of it, they don't really believe it.
As most researchers didn't believe it when they started looking at it. So it's quite okay. It's quite okay. So tell me, what do you think that the new labs are going to be doing? What are the new scientists, many of whom you have tutored, many of whom you have mentored? What do you hear what they're working on? What do you think that they're going to say what are they going to be studying? Hi, love.
I'm getting. Sorry.
What do you think is going to be published in the next 10 years? What are we going to know about? Because I know that you got your finger on it. I know you've got stuff in the labs, but we're 10 years from now. What are we going to see? What people are going to be studying? What are they going to understand better? Let's put it that way. You know?
Well, my personal thing is it's going to be Alzheimer's and Parkinson's that are really going to hit. You know, I mean, stroke rehabilitation is a huge, big deal, and not many people study it. You know, the ones that have studied it, it makes a big effect. I mean, stroke rehabilitation is always going to be a slow business, but putting light on the head significantly accelerates it. And a lot of people have strokes. You know, obviously the big one. I mean, the psychiatric disorders as well. It's just started, you know. I mean, the worst thing that happened to psychiatry was the introduction of psychiatric drugs. You know, because back in the days of psychoanalysis, at least the psychiatrists had to take their job seriously.
Yeah, I'm surprised at the number of psychiatrists that I've interviewed for this program, and their perspective and how, you know, actually seeing, you know, helping patients get better was what drove them into using photobiomodulation. So I agree with you, Doc. I agree with you. Any other closing statements before you get to your wine? I don't want to keep you from your wine.
Because it's dinner time here. You know?
Well, that's time, I know. Any closing statements, anything that you want anybody else to know, or message to the world, a time capsule here, anything?
Well, yeah, just keep on with the light. It has zero side effects, can't do you any harm, and you never know how much good it'll do you till you try it.
Thank you so much, Doc. Enjoy your dinner, whatever it's going to be. I'm sure it's going to be lovely.
All righty. Thank you, Joe.
Thanks so much.